Hi, good day, everyone. Um, I decided to go over the test questions over Chem 315. Initially, I wanted to release the result, and I felt like how the result is going to help you out. It is very difficult, and I later on think also of giving out the script, the test script. And I also felt most of you have not, quite a number, majority have not done the right thing. So how will you know that what you gave as an answer is not actually what is expected? So and the exam have started, there is not much time to come and find out and how many people will come, my schedules on the road. So I felt I need to have some analysis of the questions and the expected response so that in future you might come across something like that. It will help you in knowing where to start and how to go about addressing the questions. So the video will be a very short one. Please take your time and listen to it. Now, starting with the first question. Let me take this to here and see. Question 1 says, outline briefly how amino acids contribute to metabolism in fed and fasting state. In this question, one thing I think I was expecting is that you look at the metabolism of amino acids in the fed state. After you've eaten a food that contains a protein, there is the digestion and absorption. Yes, you have a lot of amino acids being absorbed. Now, from there, what happens? What is the fate of the amino acid after you have eaten? After eating food, there are a lot of amino acids in the system. Of course, the amino acids will be used for protein synthesis. So I'm expecting a response that talked about presence of amino acids or excess of amino acids from food can trigger protein synthesis. When enough proteins are produced, then you have excess of these amino acids, and they will be converted to glucose through gluconeogenesis. So if you like, you may decide to say this excess amino acids undergo degradation or transamination or transdeamination to form the alpha keto acids. And the alpha keto acids, some of them will be used to produce glucose through gluconeogenesis, while some will be used to produce fat, like fatty acids. And then the glucose and the fatty acid will be stored in form of glycogen or triacylglycerol in adipose tissue for energy storage. And you can even go ahead and say some of these amino acids can be used to synthesize nitrogen-containing compounds. That is in the fed state. But during fasting, when the body needs energy, and maybe there is no much amount of energy, because although there is a difference between fasting and starvation, but most important thing here is that you need to know during fasting, usually the glucose used to be low. And when the glucose is very low, there are some certain cells in the body that actually need the glucose for metabolism. And if there is no glucose, then they don't go for fatty acids, they go for ketone bodies. So of course, some of these proteins are broken down. So you have a protein degradation to produce amino acids, and these amino acids, the glucogenic will be converted to glucose and being utilized by some of those tissues like brain and red blood cells. And the ketogenic will now be used to produce the ketones. And then the ketones will be used by brain, the muscles for metabolic activity. So basically, in summary, this is what is expected of you to briefly say about this question. I learned that quite a number of you were able to say one or two things, while some were completely lost. Question 2 said, nutrient deficiency, such as vitamin B6, can affect amino acid catabolism. Provide a concise explanation how and what are the possible consequences. This is a very simple. You can remember this reaction by the right, the conversion of amino acid to a keto acid. And we said this reaction is classically catalyzed by aminotransferases. And this aminotransferases require one particular cofactor which we emphasize as pyridoxal phosphate. And this pyridoxal phosphate we said is formed from pyridoxine, which is vitamin B6. So the precursor of pyridoxal phosphate is vitamin B6. So in the absence or deficiency of vitamin B6, there is going to be an impairment in the transamination reaction by this enzyme. And of course, when there is going to be an accumulation of toxic nitrogenous substances. And one of the consequences we mentioned, most especially with regard to accumulation of this, it affects the synthesis of neurotransmitters, and that is related to neurological disturbance, and that also related to energy utilization. This leads to weakness, and quite a number of consequences we've mentioned. And basically, this is something you can summarize in 1 or 2 sentences. And this is what is actually expected of you in this question. Now, the next question, question 3, says, using any detailed biochemical reaction as an example, explain the mechanism of non-oxidative deamination. So in this question, it doesn't need you to start talking about or speaking English. Rather, I said a detailed biochemical reaction. A detailed biochemical reaction, you know, all these ones, you know the structures. Cysteine, you know the structure of cysteine. Pyruvate, you know the structure of pyruvate. And what else? Because when we say detail, at least we're expecting you to write the structure. So these are things that you can do with, you all know the structures because it involves as very, very simple amino acids. So you can put either of the one, cysteine, converted to pyruvate by desulfohydrasis, releasing an amino group, which we said deamination, and then hydrogen sulfide. Simple. Or you may decide to say serine by an enzyme dehydratase, removing ammonium to produce alpha keto acid, or threonine, or homoserine. Any one you give here, I think is you get the polymer. So as simple as that. It's not along the English, it's expected here. The question 4 says, approximately 75% of kidney stone cases are associated with calcium oxalate crystal. Explain how their formation related to amino acid catabolism. And we talked about the different pathways through which glycine can be broken down. And one of them, we say glycine is converted by amino acid oxalate to glyoxalate. And we say glyoxalate is converted to oxalate through alcohol dehydrogenase light reaction, a reaction that requires the NAD, NAD plus NADH plus H. This is classically, and we said this oxalate in the kidney can convert with calcium to form calcium oxalate, and this calcium oxalate can form a crystal, and deposition of this crystal in the kidney leads to kidney stone. And typically, when you give this reaction event, maybe put one sentence like oxalate in the kidney combined with calcium to form calcium oxalate stone deposited leading to kidney stone. I think you have actually attended this question, and there is no much thing expected. So some of you were able to, but some of you were completely off. Question 5 says complete the blank spaces in the table below and provide a detailed biochemical reaction involved in any one of the listed conditions. Condition 1 to 4. And I think I gave the conditions, and I gave at least an example of the genetic disorder, an example of although I mentioned almost all the genetic disorder, excluding the last one, which is phenylketonuria. And I also gave an example, 1 or 2 of the reaction affected, I think 1. I also gave an example of the distinct symptom and then the management. So I gave this, okay, I didn't give anything management. So I actually give this for you to have an idea of what I'm expecting. For example, in albinism, I said hydrolysis. So in neonatal tyro, in one of them, I gave an example. Is it tyrosinemia, where I said hydrolysis of, for example, let me check albinism, because let me check the question to be sure what I exactly read. Okay, in the question, I only gave the genetic disorders and reaction affected for phenylketonuria. So I gave you at least an information of what I'm expecting when you want to tell me the reaction affected. I said conversion of phenylalanine to tyrosine by phenylalanine hydrolysis. So just the reactant and the final product and then the enzyme involved. But some of you went and put in some things that I don't even understand. Probably they didn't wait that. So, and the distinct symptoms are actually some things you already know. Even though many people went ahead and put so many things that are not even related, but and as well the management which we have discussed. In most cases we talked about maybe taking low diet containing low of the affected amino acids, which are mostly the management approaches we explain. So basically these are the things we am expecting for the test, and the test was written, and I felt giving the script may not help you better than doing it this way. So please take your time, listen to this short video before you write your exams. I'm sure the response and the way the questions were answered will help you in answering those some of the similar questions that will come out during exams. I just need a very short explanation that you don't have to start giving me history of some metabolism before you come down and tell me exactly what I asked you. Just go straight to the point and answer the question. I wish you a great success in your upcoming examinations. Thank you very much.